Fine. Neural control. They have to do neural control disorders. Disorders? We did or not? No? Respiratory disorders?
No, sir.
Okay. Respiratory disorder, it is also important because question have been asked from like many time question have been asked from emphysema, and the second one occupational respiratory diseases. These were the two questions which has been asked many times in your medical examination. So before going to circulatory system, we must finish the respiratory system. The last topic, the last small topic, that is the neural control. So put the heading. Neural regulation of respiration. Neural regulation of respiration. Regulation. Right. Now let me first explain it for you. So try to understand what exactly I want to say. Look, in neural regulation, for example, suppose that, so this is our brain part. So this is our brain, and so this is the part of brain. Which part is this? This is actually hindbrain, right? This is hindbrain. Okay. So in hindbrain, we know that this swollen part is pons. We'll again discuss all these part of the brain in the next chapter, like next to last I think second last chapter. So that is the neural regulation, right? So this part is called actually pons, and this part is actually called medulla oblongata. Let me write it down here only. So just focus over here and try to understand whatever I want to communicate you. So these are the two parts. Right? So basically, this is the pons varoli, and pons varoli, and this is the medulla oblongata. Medulla oblongata. Out of this, there are two regulatory control. They are the made up of actually they are the cluster of neuron. So two regulatory controls are present over here. So one is here and one is here. And two more regulatory controls are found. So two regulatory controls are found in this part, actually pons part, and two regulatory controls are found in medulla part. Actually, we know the clearly function of those part which are found in this medulla part. Medulla. What exactly we want to we say these part? Let me make it clear. So this one, this one, and again this. So this above one, this is called pneumotaxic center. Pneumotaxic center. Pneumotaxic center. Actually, this pneumotaxic center is responsible for it primarily limit the limitation. It primarily limit the inspiration. It primarily it primarily limit the inspiration. So it is responsible for breathe in process. Inspiration. Right. Now the second one, that is the apneustic center. Apneustic center. The function of apneustic center is not well known, right? So we hardly know, but still, as what the scientist says, that I'm telling you, not well understood, well understood, but it controlled depth of inspiration. It control depth of inspiration, right? So during emergency, that depth will increase. During the normal condition, the depth will be normal. So that is the depth, like you breathe in, like you breathe in long. So that is the depth of respiration. That is controlled by these two centers. And actually, they send the information. These two centers, they send the information to this one, right? This center. They send the information to this center. Now, what we call these center, which I have shown in the green color. So let me again make you understand. Uh, yes, yes, right. So this is actually uh, this is called DRG. DRG. DRG is dorsal respiratory group. Dorsal respiratory group. Actually, uh, this is present in dorsal part, and it is the cluster of neuron. And the next one, this is the VRG. VRG. So VRG is ventral respiratory group. Ventral respiratory. So DRG and VRG. Right. Now look. So these are the this is the like these are the ribs. These are the ribs. Right. This is the backbone. Again, these are the ribs. This is the backbone. So these are your ribs. So what happened? Uh, a nerve from here, it comes to here, this part, these part ribs. And what exactly it do? It intercostal muscles contracts. So contraction of contraction of intercostal muscle. Intercostal muscle is done by with the help of by the command of this VRG actually. And when? During the expiration and inspiration both. During during expiration and inspiration both. During inspiration and expiration both. Next, one more important thing. There is another nerve. There is another nerve which come from here, right? From DRG. So it originates from DRG, but what happened? It bifurcate into two part. One part goes over here, right? And this one part, what exactly do? It controls the external intercostal muscle. External intercostal muscle contract during inspiration. Inspiration. Again, the another nerve which originate from here and it comes over here, it control the diaphragm actually. So when it control to the diaphragm, so diaphragm is also responsible for increasing the or decreasing the volume of respiratory gas. That's how the nervous system control our respiration, right? So VRG is responsible for contraction of internal intercostal muscle, right? And DRG is responsible for contraction of external intercostal muscle. That's how our respiratory respiratory system is controlled by our nervous system. So look, the main role of nervous control of respiration is of DRG only. DRG and VRG means the main role, the main center of respiration in our human brain is found in the medulla oblongata. Most generally controlled, but it is not that much important. It is not that much important center as the medulla oblongata is. Right? Is it clear to all of you? Did you got it?
Yes, sir.
Great. So please copy as it is. This is the simplest form which I have explained you, else you will get confused, right? So copy it as it is. Please let me know whenever you note it down.
Also.
Done. So everybody completed? Fine. Now the next topic is like the last topic of this uh, chapter is that is respiratory disorders, right? Respiratory disorders. So there are three disorders which are very important. Respiratory disorders. So please note it down. The respiratory disorder. The first respiratory disorder is bronchial asthma. That is bronchial asthma. Actually, bronchial asthma is a type of allergic reaction, and the allergen may be anything, right? Sometimes it takes due to dust, due to pollen and all that, right? So please note it down. Bronchial asthma. It is caused due to allergic reaction. It is caused due to allergic reaction to foreign substances that affect to foreign substances that affect to affect to respiratory tract, respiratory tract. Full stop. Allergens stimulate allergens stimulate and release the histamine. Allergen stimulate and the histamine, release the histamine from mast cell. So we have already discussed that is the animal tissue part, but still I want to remind you again. The mast cell are actually found below our skin. They are a special kind of the cell which tells us that a certain attack has been done at a certain part of the body. So whenever any antigen enter in our blood system, either from the our skin or injury or any part, right? These mast cells start releasing histamine. Due to histamine, itching takes place in our body, right? We get itching, right? Itching, that part become red. That is all caused by histamine. So mast cells are responsible for the allergy. Allergens stimulate release of histamine from mast cell. Full stop. Which have several which have several effect as as bronchial smooth muscles to contract. They have several effect as bronchial smooth muscles have to contract and cause bronchial asthma and cause bronchial asthma, comma, coughing, wheezing. What is wheezing? Can you tell me what is wheezing? Wheezing is that part when your respiratory tract get choke, and whenever a person take in or like inspire or expire, you will clearly listen the noise of like inspiration and expiration, right? So that sort of thing is called wheezing. Whenever suppose that a person, you must have seen during the winter time, during this time, uh, so when someone is taking breathing, if I have cold, right? So whenever he will breathe in or breathe out, you can listen the sound of air going in and coming out. That part is called wheezing. uh, Wheezing. Excess of mucus is secreted. Excess of mucus is secreted by respiratory tract. Next disease is emphysema, that have been asked multiple time in medical exams. Emphysema, it is also important for your school exams. Emphysema. Emphysema means inflation. Emphysema means inflation or full of air. Full of air. Emphysema is inflation or full of air. It is an it is an abnormal distension of bronchioles, abnormal distension of alveoli or alveolar sac, alveole or alveolar sac. Full stop. Many septa between alveoli. So what happens? Suppose that inflation means it got expanded. So look, suppose that these are the septa. These are the actually septa. These septa get broken. These are the septa of alveoli. When septa broken, these alveoli expand, inflate. So that is an inflation, and that's why the full of air. It seems like they are full with air, right? Many septa between alveoli are destroyed, and and much of elastic tissue, much of elastic tissue of lungs, much of elastic tissue of lungs replaced by connective tissue, replaced by connective tissue. First off, major cause of major cause of emphysema is cigarette smoking, and major cause of emphysema is cigarette smoking and inhalation of other toxic smoke and inhalation of other toxic smoke. Next point in the same emphysema, septa collapse, septa collapse, hence major area of gas exchange, septa collapse, hence major area for gas exchange is greatly reduced. Lungs remain inflated, lungs remain inflated and remain inflated even after expiration, even after expiration, even after expiration. Right? Next. The third one is occupational respiratory disorder. Occupational respiratory disorder. Occupational respiratory disorder. Right? It is caused due to it is called due to harmful substances. It is called due to harmful substances such as gas, fumes, dust, such as gas, fumes, dust. Full stop. Silicosis and asbestosis. So there are two examples, right? So the first disease is silicosis and asbestosis. Asbestosis, silicosis, and asbestosis are the common example. Actually, silicosis takes place. You must have seen that if you will walk in the market where glass cutting takes place. So whenever they rub the glass with the machine, a small particle of silica they start flowing in with the air, uh, and they goes inside their nostril and respiratory tract as well. Because of this silica, they got respiratory disease. Right, that is called silicosis. Again, there is one more thing that will it look like cement, that is asbestos. That asbestos, the worker which work in the factory of asbestos, it is used in cement as well. So the worker which you work in the factory of asbestos, they get infected with the asbestos. Asbestos goes inside their respiratory tract and causes some diseases. Right? These are the three major diseases of respiratory. So that's it for the respiration. Is it clear or not? Please tell me. This was all about the respiration. Please check the NCERT. And in case if you have any doubt in respiration part, then we'll just solve over here. Then we'll move to next chapter, that is the circulation, human circulation. Samara, all clear? Minashi, Minashi, Atif, Fazia, all clear?
Yes.
Okay, great. So now we are going to start a new chapter, and our new chapter is that human circulation, right? Human. Okay. So the next chapter is human circulation, or human circulatory, you can say human circulatory system, whatever you want to say. Just give me just. Let me open the NCERT. So the name of the actually chapter is body, fluid, and circulation. Body, fluid. The first topic is like simply let me discuss it. Okay, okay, yeah, sure, Samara. That's why yes, oxygen dissociation curve. Sure. Oxygen dissociation. Let me explain this graph. What is this graph shows actually? It shows the oxygen carrying capacity of hemoglobin actually. Respiration. Uh, respiration. Yes. Fine. This is actually oxygen dissociation curve. Look, uh, down the line here, this is PO2. That is the pressure of oxygen, right? And mmHg, right? It has been given pressure of oxygen, right? Here it is the oxygen saturation of hemoglobin, right? So here, whenever the level of oxygen or pressure of oxygen means concentration of oxygen increases. In the same way, the carrying capacity of percentage of saturation of hemoglobin increases. It keep on increasing, but at a certain point, after a certain point, like that is the that this is the pure point. After this point, it start declining, right? So this is the physiological. This is called physiological range. So after that, what happened? Uh, that the oxygen carrying saturation level decreases. So what happened? Suppose that there are 1 lakh molecule of hemoglobin are available over there. Whenever you increase the oxygen concentration, more and more hemoglobin molecules increases. So when all the hemoglobin molecule will get engaged with the oxygen molecule, then there will be no more capacity to carry the oxygen, right? So this curve will become straight, and after a particular level, after a critical level that has been given over here, this 40, after a particular level, this curve will not go up, or oxygen saturation or hemoglobin saturation will not increase. That is the curve. Clear, Shamara? Is it clear? Please tell me.
Yes, sir, clear.
Okay, now let's start with the circulation part, right? So before going deep into the circulation, mm, uh, I just want to discuss some like very basic kind of thing. So the first important topic is blood, right? So what is blood actually? What is the composition of blood? That is actually my question. What is the composition of blood, right? So let's discuss what is the composition of for sake of example, please try to understand. For sake of example, suppose that this is a glass. This is a glass. This is a glass, and this glass is filled with blood. So this glass is filled with blood, right? So it is considered blood. No, no, not blood. Okay. So suppose that it is filled with blood. What will happen after a certain time? Like after a few minutes, you will see some drastic change, right? So after a few minutes, what will happen? Uh, you will see. So how many minutes after, if it is a healthy person, then after 7 to 20 minutes, not more than 20 minutes. If there is more than 20 minutes, mean the person is diseased. Right? Okay. After 7 to 20 minutes, you will see there are some changes. What are the changes? What are those changes actually? Then you will see that there is like certain clot-like structure. So this is a basic clot-like structure. You will get there is a thick uh, part which is made up of the blood, and along with this one, there is something yellow type of there is something yellow type of liquid as well, right? So this solid part get uh, separated with the liquid part, right? What we call this solid part or what we call this liquid part actually, right? So this is this slightly yellow liquid, right? This is yellow, slightly yellow liquid, and this is dark red solid clot, right? This part is actually called blood clot. Blood clot. This part is called blood clot. And the above one, this is called actually blood plasma. Now I have a question, and that is very basic question, and actually I'm asking this question to you because it also have been asked in your medical examination. What is the percentage of plasma in blood? Come on, tell me. Samaira, Maziha, Minashi, all people, tell me. What is the percentage of plasma in blood? The process is called blood clotting or blood coagulation, right? This is the blood clotting process. Blood clotting. So I have given you one more data. How many? What is the time of clotting? In a healthy person, the time of clotting should be 7 to 20 minutes, not more than that. So blood clotting, or we also called it blood coagulation. Come on, tell me what is the percentage of plasma in blood? Samira, what is percentage?
So don't know.
Atif, any idea? No.
Was
Look,
it 75%?
75%. Uh, okay. No, it's not
No,
75.
sir, 55%. 55%.
Yes, very good. Minashi, yes, you are also good. 55%. Very good. So the 55% part is made up of 55% part is made up of blood plasma, and rest 45% part is made up of blood clot. Again, this blood plasma, it have 90% of water, then the rest of the 10% is, that is, mineral, mineral, fat, protein, hormone, sugar, right? So this is the actually composition of blood plasma, right? And out of this 45 percent, the major composition is RBC, WBC, WBC, uh, RBC, then WBC, then platelet, platelet, then fibrinogen protein, fibrinogen protein, and one more, thrombin protein, thrombin protein, right? So these are the actually composition of blood. Please note it down. And one more important thing I want to share with you, there is a hormone which remains dissolved with the blood and would do not allow blood to clot inside our body. What is the name of that hormone? That is heparin. So there is a hormone. Name of the hormone is heparin. Heparin hormone do not allow blood to clot inside our body. So whenever blood come out of the body, it stop working, right? So heparin is also called anti-clotting agent. It is also called anti-clotting agent. So heparin is called anti-clotting agent, right? Note it down, please. Or it is also called anticoagulating agent. Anticoagulant. Please note it down and let me know.
Done, sir.
Okay, great. So please note it down some point in the bullet form, right? Water is about water is about 60% of body weight, or water is about 60% of body weight, or 42%, no, 42 liters, sorry, 42 liter in human, 42 liter in human of average weight 70 kg, human of average weight 70 kg, full stop. And increase in the percentage of fat, and increase in the percentage of fat leads to decrease in leads to decrease in leads to decrease in the water of body. Next, intracellular fluid, that is with protoplasm, right? Intracellular fluid. So intracellular fluid, intracellular and fluid have large amount of potassium. Intracellular fluid, intracellular fluid have large amount of potassium and phosphorus, potassium and phosphorus in form of iron, while extracellular fluid, while extracellular fluid, extracellular fluid have sodium and chloride, sodium and chloride, sodium and chloride, iron as an important constituent. Next point.
As an
As a important constituent, as a important constituent. Next point. Blood forms blood forms 30 to 35% of extracellular fluid. Extracellular fluid. So ECF. This is called ECF and this is called ICF. ECF and ICF. Right. So blood constitute blood constitute 30 to 35% of ECF. Full stop. An adult person, an adult person of 70 kg weight An adult person of 70 kg weight contain 5.5 liter of blood, 5.5 liter blood of of pH value 7.4 of pH value 7.4. These were the actually general points. Now let's discuss. First, we'll write down about the plasma. We'll discuss about the plasma. Then we'll write one by one that RBC, WBC, and the rest of the thing. Clear, right? So next is that is the composition of blood. We'll study one by one. First, we'll study plasma, then the rest of the thing. So the next one is next is composition of blood plasma. Composition of blood plasma. Right. So please start. So first is plasma. Plasma means blood plasma. Plasma is alkaline. Plasma is alkaline, non-living. Plasma is alkaline, non-living, and constitute about 55% of blood. Next, water alone forms water alone forms water alone forms 90 to 92%. Water alone forms 90 to 92% of plasma. Water alone forms 90 to 92% of plasma. Next point. It contains mineral, nutrient, and protein. It contains mineral, nutrient, and protein. Next, it contains 6 to 8% of protein plasma. It contains 6 to 8% of protein plasma, which is very important, which is albumin and globin. Albumin and globin. Let me discuss here the role of actually albumin that have been asked multiple times in medical examination. So actually, pH value of the blood is 7.4, right? That is the pH value of blood. Since morning to the night, we eat and drink each and everything that is the 99.9% is acidic because all the tasty things are acidic. Even the water which we drink, that is not neutral. The water which we drink, that is actually what? Acidic, right? So everything is acidic, either milk or sugar, fat, protein, all things are acidic. So, but still the pH value of blood remains 7.4, means it remains basic. How does it take place? Actually, albumin protein it work as buffer solution. It do not allow uh, the change in the pH value of blood. So albumin protein is responsible for maintaining the pH value of blood. Please note it down. albumin protein work as buffer solution. Albumin protein work as buffer solution, buffer solution, and it maintain buffer solution, and it maintain the pH value of it maintain the pH value of blood. Next, lysozyme lysozyme and lysozyme and properdin lysozyme and properdin are also found in blood plasma. Lysozyme and properdin are also found in blood plasma. Let's stop. They naturally they naturally destroy bacteria, virus, and toxic substances. Bacteria, virus, and toxic substances. Full stop. Anticoagulant heparin, anticoagulant heparin, anticoagulant heparin is also found. Anticoagulant heparin is also found in blood plasma. Next, it also contain clotting factor. It also contain clotting factor in inactive state. And put a note over here. Even this question have been asked in medical. Note here. First, I want to ask all of you, if any one of you have as a, what is serum or what is blood serum? What is blood serum? Can you tell me what is blood serum? Note it down. The plasma without clotting factor. The plasma without clotting factor. Plasma without clotting factor is called blood serum. Plasma without clotting factor is called blood serum. Uh, okay. Now come to the that is the composition part, RBC, WBC, and all that, right? So in blood composition, so serum, next is blood composition. So, we see. Platelet. Look, so the first is RBC. RBC is called erythrocyte. erythrocyte or red blood corpuscles. Red blood corpuscles, right? So we do not use term cell, red blood cell. We call them corpuscles. Why? Because there are many cell organelles are missing. Even in RBC, nucleus is also missing. So there are many organelles which are missing over here. That's why we do not use cell term. We cannot give them a status of cell. We call them that is blood, that is corpuscles, right? So that's why we use this term. So please start noting down RBC. So RBC is actually of oval shape, right? So let me incorporate a diagram over here. Then we'll go ahead. Look, these are the actually cells, and every cells have its own shape, its own structure. So this is the red blood cell, means red blood capsules. Actually, the wrong term is cell. Cell is wrong term over here. So please start noting down. So first heading is RBC. A normal man and woman have 4.5 to 5 million, 4.5 to 5 million RBC per cubic mm. RBC per cubic mm. Next, abnormal rise in RBC. Abnormal rise in RBC is called polycythemia. Abnormal rise in RBC is called polycythemia, and decrease in number of RBC, decrease in number of RBC is called erythrocytopenia. Erythrocytopenia. Next point. RBC do not have its nucleus. RBC do not have its nucleus, and it is biconcave in shape. So, can you tell me there are some animals. In fact, there are some mammals which have nucleus in their RBC. There are two mammals which have nucleus in their RBC. Please tell me the mammals which have nucleus in their RBC. Note it down. Camel and llama. Camel and llama are exception in exception in mammals which have nucleus in their RBC, which have nucleus in their RBC. Full stop. The size of RBC is 7 to 8 micrometer in diameter. The size of RBC is 7 to 8 micrometer in diameter. Next, they have red color, they have a red colored pigment called hemoglobin. They have red color pigment, and we represent with this uh, alphabet, capital H, small b. Hemoglobin is made up of globin plus heme. Hemoglobin is made up of globin plus heme. That means iron group. Globin plus iron group. 100 ml of blood. Next point. 100 ml of blood of individual. 100 ml of blood of individual contain about 12 to 16 gram 12 to 16 gram of hemoglobin. 12 to 16 gram of hemoglobin. So, very easy kind of the question, or not that much easy. It's a bit smart question. So I have seen a question, and even I want to discuss you. First, you tell me, in fact, inside mother's womb, after if you will see, uh, after the 2 month pregnancy, the heartbeat comes in fetus, right? Heart start pumping, and the fetus or human fetus have blood inside its body. So where it gets blood, from where a baby which is developing inside the body of mother get its blood formation takes place, or they take blood. Any idea? Okay, so keep in mind, uh, an infant or baby do not get blood from mother. Actually, blood transfusion do not takes place through placenta. If accidentally blood transfusion takes place through placenta, the infant or baby, it gets that a disease called pneumonia, right? And actually, in the body of even they do not have bone marrow at the infant stage. Actually, they do not have bone. It is made up of all made up of cartilage. So, in an infant, actually, the formation of blood takes place in liver, right? And whenever a baby born in this world, after the birth, uh, it start feeding on the milk, and the cartilages becomes bone, and bone have bone marrow, and hence the production of RBC takes place, right? Please note it down. RBC is produced. RBC is produced in liver. RBC is produced in liver at embryonic stage. RBC is produced in liver at embryonic stage. Embryonic stage, right? Next, uh, but from birth onward, but from birth onward, formation of but from birth onward, formation of blood takes place. Formation of blood takes place in bone marrow. Next, hemoglobin, hemoglobin combined with oxygen, hemoglobin combined with oxygen and gives rise to gives rise to oxyhemoglobin. See, hemoglobin, right? Oxyhemoglobin. Oxyhemoglobin. So please note it down. So one more important point: lifespan is lifespan is 120 days. And what will happen after the death? After the death of RBC, they goes into an organ called spleen, right? Spleen. So spleen actually it is called all dead RBC goes to spleen and they are broken over there, right? So spleen is that's why spleen is called graveyard of RBC. Please note it down. Spleen is called graveyard of RBC. Graveyard of RBC, right? Give me just 1 minute, right? Just a minute. I'll be back in 1 minute. Yes. So again, let's start. So we were in RBC, right? So RBC clear? Is it clear to all of you?
Yes, sir.
Okay. Then you will see there are different type of the WBC. I will tell you all the type of WBC. So spleen is called graveyard of RBC because all, if you want, please note it down. Spleen is called spleen is called graveyard of RBC because spleen is called graveyard of RBC because after the death because after the death because after the death all of the because after the death all of the RBC goes to all of the RBC goes to spleen for destruction. So practically, I want to tell you that there are approximately 80 lakh RBC died every day in our body. So if these dead RBC will keep on accumulating in our blood, they will make the blood dirty in few days, right? So what happened? We have got a special type of the filter in our body that is called spleen. So when blood passes through spleen, what they do, they capture the dead RBC, and they break it down, and they utilize the important part, recycle the important constituent of the RBC, and the wasteful material is excreted out, right? So that is the function of. And one more important, the spleen is also called blood bank. That is also a question. That is also a question. So you can write down, spleen is also called blood bank because because extra RBC because extra RBC are stored over here. Extra RBC are stored over here. Fine. Look. Further, if we talk about WBC, so next is WBC. Let me tell you, WBC are of many types. As you will see over here, that WBC, the second name of WBC is that is called leukocytes, right? So these leukocytes are further many types: monocyte, lymphocyte, eosinophil, basophil, and neutrophil. These all are type of the WBC. What is the difference and all that we are going to discuss few minutes later. But before that, we need to understand the general characteristic of WBC. So put the next heading, WBC. So next heading is WBC. Next is WBC or leukocyte or WBC or leukocyte. WBC or leukocyte. Please note it down. WBC. So first point, their number varies. Their number varies from 6,000 to 8,000, 6,000 to 8,000 per mm cubic, 6,000 to 8,000 per mm cubic of blood. Full stop. Ratio of, next point, ratio of WBC to RBC is 1:600. 1:600. Full stop. Rise in WBC termed leukocytosis. Rise in WBC termed leukocytosis, right? And it shows acute bacterial infection. And it shows acute bacterial infection. So whenever the bacterial infection will take place, right, the number of leukocytes or number of the WBC will increase because their function is to defend our body from diseases. Next, but so this is the normal increase in the level is called that is leukocytosis. But abnormal increase is called blood cancer. That is called blood cancer. That is called leukemia actually, right? So please note it down. Abnormal. Next point. Abnormal increase in WBC leads to leukemia. Abnormal increase in WBC leads to leukemia. Full stop. It is also called blood cancer. It is also called blood cancer. Next, fall in WBC is called fall in WBC is called leukopenia. Fall in WBC is called leukopenia. Next, shape of WBC, shape of WBC changes, and sometimes it is rounded or irregular. Sometimes it is rounded or irregular, but they have nucleus. But they have nucleus. One more important thing which I want to discuss with you, that what is the benefit of being changing their shape? So what benefit they do have? Look. So suppose that this is our blood capillary. This is blood capillary. This is blood capillary. Inside blood capillary there are RBC and even WBC too. But yes, WBC can change their shape. Because of this their property, they can squeeze out of here, right? They will squeeze out. These are not perfectly uh, like impermeable. Actually, they have some spaces, and from these spaces, these WBC come outside of the capillaries, and here they are able to fight, right? Fight with the disease and kill them, right? Please note it down. Change in shape enables them, change in shape enables them to squeeze out, to squeeze out, to squeeze out of blood capillaries into tissue, blood capillaries into tissue. Next, size in size is 10 to 20 micrometer. Next, formation of formation of leukocyte is formation of leukocyte is called called leukocytosis or or leukopoiesis or leukopoiesis or leukopoiesis, right? Now I am going to show you the type of WBC. Further, there are many type of the WBC, and every type have its own function, right? So let me tell you the type of WBC. Oh, very large. Let me make it smaller. Look, Please try to understand and let me explain you. Look, so WBC is also called leukocytes, right? WBCs are also called leukocytes, and leukocytes have been further divided into two parts: granulocyte, agranulocyte. So agranulocyte, they do not have granule, do not have granule in cytoplasm, right? Granulocytes, they have granule in, they have granule in protoplasm. That's the only difference. So further, agranulocytes have been divided into two parts: lymphocytes and monocytes. Right? Lymphocytes, they may be B or T lymphocytes. So there are two types of the lymphocyte: B lymphocyte. So B lymphocytes are actually produced in liver, right? And T lymphocyte is produced in thymus gland, right? That is the difference. P and T, non-motile and non-phagocytic. They are non-motile and they are non-phagocytic, right? They produces antibody and destroy the microbe and toxins and tumor cells, right? So the question it has been this question have been asked that which of the cells responsible for destroying the tumor cells, right? So these kind of like lymphocytes are the type of WBC which destroy the cancer cell. This have been asked in your medical examination, right? Next, their number they are 20 to 25% of WBC. Next, lifespan is few days to few years, right? So from few days to year, it's not fixed. Again, monocytes. Monocytes in a largest WBC. The question have been asked. Largest WBC. They are motile, phagocytic. They engulf bacteria and cell debris. 10 to 20 are lifespan. Again, this granulocyte have been divided into, look, this granulocyte have been divided into three categories: eosinophils, basophil, and neutrophils. Eosinophils increase in allergy, right? They increase in allergy and their number increases. And asthma, hay fever. So asthma and hay fever. They help in dissolving the blood clot, right? Non-phagocytic. Similar like lysosome, they destroy the protista, right? They also have capability to destroy protista. Again, basophils. Basophil release of heparin, serotonin, and histamine, right? And least in count, right? So they are least in count. Their number of least, right? Again, neutrophils. Most numerous. This question have also been asked 60 to 65%. So they are the most numerous of WBC. Out of uh, out of all WBC, the neutrophils are 60 to 65%. They eat harmful germs, and that's why phagocytic. So they are phagocytic in nature. Is it clear? Is this chart is clear?
Yes, sir.
Okay. Take the screenshot first. Then we'll start with the next. Okay. Now the next one is platelet. Platelet. Platelet is also called thrombocyte, right? So they have one more technical name that is called thrombocytes, right? So start writing down thrombocytes. Thrombocytes are blood platelet Normally, their number varies from 1,50,000. Normally, their number varies from 1,50,000, 1,50,000 to 3,35,000. 1,50,000 to 3,50,000, right? Per mm cubic. Per mm cubic. Per mm cubic. Right? Next point. Increase in number is increase in number is called thrombocytosis. Increase in number is called thrombocytosis. Thrombocytosis. Next, decrease in number is called thrombocytopenia. Decrease in number is called thrombocytopenia. So, thrombo decrease in number is called thrombocytopenia. Next, they are rounded, oval, or disc shape. They are rounded, oval, and disc shape. Next point. They are colorless. They are the colorless. That means they do not have any pigment. Next, what is their function? What is their importance? So if you talk about the importance, they are responsible for blood clotting. So please note it down. Platelet formation takes place. Platelet formation takes place. Platelet formation takes place in bone marrow. In bone marrow. Full stop. Next one. Lifespan is about a week. Lifespan is about a week. Next, platelet help in blood clotting. Platelet help in blood clotting. Clear or blood capsules clear? Now the next topic is blood clotting. Right? Right? Sorry, not blood clotting. That's a blood group. AB, ABO blood group. What is blood group actually? Right. So let me tell you the blood group. Blood group. ABO blood group. Right. So the ABO blood group was discovered by ABO blood group was discovered by Ladsteiner. ABO blood group was discovered by Ladsteiner in 1900. AB was AB blood group was found out by AB blood group was found out by Ladsteiner. AB blood group was found out by Di Castello and Steny. Di Castello and 10E. The ABO blood group is determined by ABO blood group is determined by gene I and I denote for isoglutamine. Isoglutamine, right? So that is I gene. Iodine determined a person will have A blood group, B blood group, or ABO blood group. So look, before going deep into blood group, I want to tell you something basic. So what happened? Every RBC have, suppose that this is a RBC. This is RBC. So external layer of RBC will have, or membrane will have, those kind of the protein, right? This protein, it work as antigen, right? It work as antigen, right? So what is antigen, by the way? What is antigen? Please tell me. Samara, Fazia, tell me, what is antigen? Hey, come on. Atif, what is antigen? You must have listened this term frequently. No one? So please note down. First note down the definition of
So
antigen.
is it like a protein?
It may be a protein. Look, Atif, anything whichever enter inside our body, it is not naturally part of our body or blood, right? And if it enter inside body, and our WBC will start producing the antibody against it, then this is antigen for that. For sake of example, Atif, if suppose that I have allergy with dust, so dust is antigen for me, right? Whenever dust will come goes inside my body, so that will uh, 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 start working as allergen, right? So I will have some fever, sneezing, and coughing and all that, right? So antigen maybe it's not fixed. Suppose that you have allergy with the smoke, I have allergy with the dust. So I am allergic with the dust, but I am not allergic with the smoke, right? So antigen, the thing which is antigen for you is not for me, which is antigen for me, which is not for you. So the definition of antigen is not fixed, but in general, like they are the um, a type of chemical. Please note it down. Antigens are a type of substances when they enter when they enter inside our body. Whenever they enter inside our body start producing body start producing antibody against it antibody against it. This is called antigen. So suppose that a person have blood group A, or a person have blood group B, AB, or O. What does mean? It means the person will have some antigen. So person will have A antigen. So that's why we kept the name blood group A. The person and this blood group B. So antigen is B, and AB antigen is AB, and here no, right? So here no antigen. So antigen is not found in uh, this O blood group. So that's why they are universal donor, right? Again, if we'll talk about the antibody, antibody. So out of this, in blood group A, the antibody will be B. In blood group B, the antibody will be A. In blood group AB, the it will have AB antigen, and in O there is no antigen, but there is both of. Sorry, just a minute. Uh, Antibody. Sorry. Uh, just yes. So in AB blood group, right? Here in AB blood group, they have antigen A and B both, but here no antibody, right? So that's why AB is universal acceptor, and this O group, this is universal donor, universal donor, right? So these are the blood group. Clear,
Yes,
Mazia?
sir.
Okay, great, great. Mazia, clear? Look. Yes, what is the positive scenario for positive and negative? Well, and you haven't asked me. I was expecting that question from your end that somebody will ask me, but no one have asked. Just a minute. What is positive and negative blood group? So I will tell you next page. Positive and negative blood group. So how does some blood group become positive? How does some become negative? So what does mean by the positive and negative blood group? Let me tell you. Look, there is nothing much more difference between the positive and negative than know what is the negative. Look, this is please try to understand. Be focused over here. This is blood capsules. So on the blood capsule, there may or may not be covering of protein. So suppose that this is the covering of protein covering, and this is called actually RH protein. RH protein. Actually, these all the works or experiment are done on a monkey called rhesus monkey, right? So that's why the RH term came over here. Rhesus monkey. Rhesus monkey, right? So if RH factor is present in any blood group, right? RH factor is present. So suppose that RH factor is present, then blood group will be positive. And suppose that the RH factor is not present. RH factor is not present. Then the blood group will be negative. Is it clear? And this protein work as antigen. Is it clear or not? And tell me, suppose that a person have A blood group, and we give him B blood group, what will happen? What will happen? Yes, Samara, what will happen? Atif, what will happen? Person will have A blood group. We just applied him B blood group. The person will die. Why? The different blood group, inappropriate blood group, so blood group A and blood group B, right? So both of the capsules they joined, and this is called agglutination. Agglutination. This reaction is called agglutination. So agglutination will take place if two different bloods will be mixed, right? So please note it down. RH factor. RH factor was discovered by Ladistiner. RH factor was discovered by Ladistiner. RH factor was discovered by Ladistiner. Right? RS factor was discovered by Ladistiner in 1940. In 1940, in the body of rhesus monkey. 1940, in the body of rhesus monkey. Next point. Uh, there is one question. Suppose that mother is Rh negative and her baby is Rh positive. So what will happen in the body of mother? Please try to understand me. Suppose that a mother is Rh negative and he got a baby infant and it have not born. So in the body of baby, the blood group is positive and the mother is negative. Then what will happen? Mother's body will start making antibody against that fetus. So what will happen? That first fetus will survive, but it will have uh, 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 what to say? It will have some pneumonia kind of the thing, right? After, what will happen if the second fetus develop inside the mother's body and that is also RS positive, that won't survive because mother's body will destroy the uh, 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 destroy those RBCs, right? So please note it down. If the fetus inside mother is RH positive, if the fetus if the fetus inside mother is RH positive and mother is RH negative and mother is RH negative, the first baby will survive and and second baby will die and second baby will die due to HDL, due to HDL, that is homeolytic disease of newborn. Homeolytic disease of newborn. Hemolytic disease of newborn. Clear or not? Tell me, guys, is it clear?
Yes, sir.
Clear, sir.
So, The next topic is uh, blood. What is lymph? Lymph. I think it should be lymph. Let me check. Give me one second. Next topic will be lymph, if I'm not wrong. So the next topic will be blood clotting, right? So next topic is blood clotting. We will start blood clotting in next class, right? Because today we won't be able to complete it. It's a bit bigger topic, right? So blood clotting will start in next class. So put the heading blood clotting and have a glance whatever we have studied today, and let me know if you have any confusion on that. Have a glance and let me know if you have confusion with that. All clear? Tell me, I'm waiting for your response.
Yes, okay.
Clear? Okay. So let's meet in next class, right? And in next class, I'll start with the blood clotting. We'll do blood clotting. Then I will explain the lymph, and then we will start the double circulation and circulatory system. In fact, most probably, not sure, most probably we'll finish this chapter in next class, right? Okay, guys. Right.
Okay,
So
sir.
see you guys. Bye bye. Good night. Take care. See
Thank
you
you,
in
sir.
next class.